So it's my great pleasure to introduce our next speaker, Chengran Xu, who is going to be speaking to us about single-cell transcriptomics of cell lineage differentiation during hepatogenesis. Dr. Xu received his bachelor's degree from Fudan University and his PhD from Peking University, and went on to postdoc at the University of Pennsylvania. So since 2013, he's been back in China at the School of Life Sciences at Peking University, and his career is really very focused on understanding cell differentiation pathways. Dr. Xu, how are you going? You're ready? I'll hand over to you. Thank you. Yes, thank you very much.
Okay, thank you, Christy. And thanks, Guoji and Yush for inviting me to this virtual meeting. So I will present our recent work on single-cell analysis of hepatic lineage differentiation during the liver development. So the liver is the largest solid organ in our body. So the hepatic lobule is the liver's functional unit consisting of a polytroid, a central vein, and the hepatocyte arranged in linear cord between capillary network. Hepatocyte make up about 80% of the liver's mass and perform a variety of functions, including the protein synthesis, carbohydrate and lipid metabolism, and detoxification. And hepatocytes also synthesize and secrete bile. So bile is collected and transported by biliary ducts, also called cholangiocytes. So in the liver, hepatoblast and cholangiocytes are of anodome origin, while most of other lineages are derived from mastodon. So due to the important functions of the liver, the hepatic diseases, including fibroblast, cirrhosis, and liver cancer, seriously threaten human health and life. So the liver transplantation is a common treatment for patients with severe liver failure. However, the amount of donors is insufficient. So an alternative way to cure the liver diseases is to differentiate the embryonic stem cell into liver cells or liver organoids for cell-based therapies. So this induction process generally mimics the process of hepatic lineage differentiation in vivo. The liver is considered as the anodome organ. So the development of a liver goes through a serious stage of hepatic specification and the liver diverticulum and the liver bud growth and the hepatic lineage differentiation and the maturation. So the process of the liver induction have been well studied. The earliest hepatic progenitors are specified from a liver domain of fungal anodome at 8.5 days of mouse embryo. So this process is regulated by several cell signals and a series of transcriptional factors. The earliest generated liver cells called hepatoblast, so which are bipotent and can give rise to hepatocytes and cholangiocytes. So some key transcription factors and the signaling pathways have been reported to regulate the hepatoblast differentiation. Although we knew that hepatoblast can generate two hepatic lineages, however, we do not know whether they are stem cell or progenitor cells with different states in the fetal liver, and how these stem cell or progenitor cells differentiate into specific hepatic lineages. To address these questions, we use a single-cell RNA-seq to trace the developmental pathways of hepatobiliary lineages. We isolate the fetal livers at the time point from E10.5 to E17.5. So first, we figure out the conditions for FACS to enrich the hepatobiliary cells from the fetal livers. So we sorted the cells populations and confirmed that this cell population by RT-qPCR of the hepatobiliary marked genes. Next, we did single cell analysis on these sorted cells. We use SmartSeq2 method. So in the PCA plot, the most of hepatic cells obtained from various stage were located on a line along PC1, and some cells form a distinct branch along PC2. So the cells were divided into two groups. So based on the expression patterns of marked genes, we found that the group 1 cells contained hepatoblast and hepatocyte, and the group 2 cells include cholangiocytes. So we circled the distributions of hepatoblast, hepatocyte, or cholangiocyte of each developmental stage. So on the hepatoblast to hepatocyte path, so the cells from the same developmental stage were clustered together, showing homogeneous at the transcriptomic level. So on the contrary, the cholangiocyte at the same developmental stage showed a broad distribution, suggesting they were heterogeneous. So during the developmental process, the newly generated cholangiocyte will continuously branch from the PC1 line. So during the hepatoblast to hepatocyte development, we identify a group of genes whose expression level gradually increased from E10.5 to E13.5. So gene ontology analysis shows that the enriched terms were mainly associated with hepatocyte function, such as metabolism and detoxification. So this data indicate hepatoblast specify into hepatocyte immediately after their differentiation from anodome progenitor. On the contrary, the hepatoblast to cholangiocyte development is accompanied with a dramatic transcriptomic change and associated with many signaling regulations. So we also found the expressions of many transcription factors that started to increase at the beginning of the hepatoblast hepatocyte development process. So in contrast, transcription factors that were upregulated during hepatoblast to cholangiocyte transition showed little or no expression in hepatoblast. So therefore, our findings support that hepatoblast to hepatocyte differentiation follow a default pathway, while hepatoblast to cholangiocyte transition is a directed process. So cholangiocytes were further divided into three groups, which represented the cell in different mature states. So on the PC plot, the cholangiocyte maturation mainly occurred along this PC2 axis. So we also identify the quiescent and the proliferative cells based on the expression patterns of cell cycle-related genes. So interestingly, the percentage of the proliferative cell increased as cholangiocyte developed from stage 1 to stage 3, while during hepatoblast to hepatocyte development, the proliferation rate steadily decreased. So immunostaining of marker of proliferative lithium cells K967 also confirmed our finding from the single cell analysis. So although several cell signals regulating hepatoblast to cholangiocyte differentiation have been identified, so we know little about the signal, the regulating cholangiocyte maturation. So we compare the more mature stage, stage 3 cholangiocyte with a less mature stage 1 cells, we found that stage 3 cells enriched PKC, ERK, MAPK pathways. So immunostaining confirmed at a higher level of phosphorylated ERK in the more mature duct cell. So we hypothesized that MAPK ERK pathway promote the cholangiocyte maturation. So this is the MAPK ERK signaling cascade. TPBB is a small chemical molecule to activate the protein kinase C function, but U0126 is the inhibitor to a downstream factor MEK in this signaling pathway. So to test whether MAPK or signaling pathway to promote the cholangiocyte maturation. So we treat the liver explant at E12.5, the time before cholangiocyte differentiation with TPPB U0126 and both. And the single cell INSIC shows that TPPB treatment indeed promote the cholangiocyte maturation. Therefore, in our single cell study, we discovered a precise hepatoblast differentiation pathway. So our data does not support that there's a steady stem cell population of hepatic lineage in the fetal liver. However, due to the small number of cells in this study, we cannot rule out that there may be some kind of stem cell, but were not detected. In addition, we do not know whether this default directed path is conserved during the human liver development. To solve these problems, we need to comprehensively profile human and mouse fetal livers. So we collected the whole human and mouse fetal livers at the various developmental stages for 10x genomic single-cell RNA-seq analysis. So the first round of t-SNE analysis identified 13 major cell lineages according to the transcriptomic similarity. So we clustered all these cells lineages into 4 major cell families: the anodome-derived lineages, erythroid lineages, and other 2 lineages. So in this talk, I still focus on the anodome-derived hepatobiliary lineages. If you are interested in other lineages, please refer to our paper, which has been online. So we found that the markers like STLK1 and CD13 used to label hepatic cells in mice were also expressed in humans. And we also identified 2 more cell surface markers for both species. And we found FGB gene was exclusively expressed in the hepatoblast in the early embryonic liver. So in the field of the liver development, people lack effective mass to trace and manipulate hepatobiliary lineages at the hepatoblast stage. So we therefore generate a transgenic mice strain expression inducible Cre recombinase under the control of FGB element. So this mice was crossed with a RUSA26 strain. The pregnant mice was induced to express Cre at E11.5, and the embryos were analyzed at 17.5. So we see that TD tomato signals were only detected in the liver, but not in other major organs. So flow cytometric analysis of liver cells showed that TD tomato marked most of the hepatocyte and the cholangiocyte at E17.5. So therefore, the FGB CRE-ERT is an efficient tool for tracing and genetic manipulation of hepatoblast. And on the t-SNE plot, the hepatoblast hepatocyte at the same development time was generally classed together. However, in the early stage, but not late stage, of both human and mouse livers, we saw a small cell cluster. There's a class shown in green. So this cluster separated from the main cluster. So this small cluster of cells express many methylome-related genes. Very interesting. So such as ID3, COL1A, and HAN2. So we named these cells ID3 positive hepatoblast. And we found that NKM1, so it's highly expressed in ID3 positive, but not ID3 negative hepatoblast. Because we can use the NKM1 to sort the both ID3 positive and ID3 negative cells for single cell culture. So both ID3 positive and ID3 negative hepatoblasts could give rise to hepatocytes and cholangiocytes. So this is a single, show that's from a single clone, show that both ID3 positive and negative are bipotent. So therefore, so we identify a novel ID3 positive hepatoblast subpopulation, although the origin of this subpopulation need to be determined. We performed the PCA of sampled hepatoblast and hepatocyte with all detected cholangiocytes. So in the PC plot, we can see that human hepatoblast also follow a default path to generate a hepatocyte and a directed path to differentiate into cholangiocyte. So ID3 positive cells and ID3 negative cells located together or overlapping along the PC1. So therefore, the hepatoblast differentiation pathway between the species is conserved. So on the PC plot, we also found that at an early stage of liver development in both humans and mice, hepatoblast and hepatocytes display a nearly homogeneous distribution. However, in the human liver, the distribution of developing hepatocytes gradually expanded along the PC2. Further, we found that in human hepatocytes, the cells can be divided into two groups. One group have highly expressed VTN genes. We name this group cells VTN high cells, and the other group named VTN low. So differential gene expression and geo analysis show that VTN high and VTN low cells, we found that the VTN high cells highly express the gene associated with the liver functions, including the detoxification and organic acid metabolism, while genes highly expressed in VTN low was associated with growth and increases in cell mass. To validate the heterogeneity of the developing human hepatocytes, we used the in situ hybridization to examine the expression pattern of VTN genes and a VTN low highly expressed gene IPL13 in the fetal liver. In the 19 weeks of human liver, so VTN positive cells and IPL 13 positive cell hepatocytes form the separate cell clusters and present a mutually exclusive spatial distribution. So we then attempt to use our findings to in vivo to optimize the process of in vitro stem cell differentiation into hepatic cells. We collaborated with Dr. Xin Chun at the Shanghai Institute. We use a human anodome progenitor cell line as a starting cells to generate the hepatic cells at a large scale. Because the differentiation from hepatoblast to hepatocyte is a default process, we impose less interference on this process. On the contrary, to produce a cholangiocyte, we applied more strict treatment to this induction process. And single cell analysis shows that in the EP cell induction system, hepatoblast also follow a default and a directed path to differentiate into hepatocyte and cholangiocyte, respectively. So therefore, in both humans and mice, we identify novel markers of hepatic lineages and a novel ID3 positive hepatoblast subpopulation. We found that a default directed hepatoblast differentiation pathways are conserved. And we identify the heterogeneity of human fetal hepatocytes. Our in vivo study can be used to guide the in vitro stem cell differentiation approach. More importantly, our studies establish a model system to study the regulatory mechanism of default versus directed cell lineage differentiation from multiple aspects. Finally, I want to thank Li, Xin, Wei-Lin, and Yan-Chun for their major contribution to this work, and thank the members from our lab, our collaborators, and others who provide assistance to our project. Thank you all, and I'm glad to answer questions.
Thank you very much, Dr. Xu, for a really interesting talk. Can I ask you to stop sharing your screen, please? While you're, thank you very much.
Okay.
So I'll invite the audience to submit questions via the question and answer box. And in the meantime, I would like to know how mature the cells become in the in vitro culture compared to the the in vivo cells that you were profiling, do they reach a good level of maturity, or are they still quite early?
Okay, so the in vitro induction system, we use single cell study, just study the early stage, just from hepatoblast to other two lineages. And all these cells shows, we culture longer, they become more mature. And if we transplant into the mice, it works well on the some liver function deficient mice. So but you just compare that in vivo, our in vivo data with the in vitro induced hepatoblast or hepatocyte early stage, but there's still the big difference at the transcriptomic level.
Yeah, yeah. It doesn't surprise me, but it's very interesting, isn't it? Do you see any evidence of de-differentiation and then re-differentiation, or is it all one way? You're describing a hierarchy of differentiation. I'm wondering if there's any evidence of the hepatocytes or the cholangioblasts reverting in culture.
In culture, so I think in culture we found that just cell differentiation just go just the one direction, not we didn't see that cells de-differentiate. Because maybe that happens in the regeneration process. In this model probably that happened.
I see. Thank you very much. So I'm going to thank all of the speakers in this session and hand the discussion back to Dr. Tang.
Yeah, I want to thank also all the three speakers for the great talk, and also thanks the people, the audience just asked very nice questions. Okay, so I think the session is finished. So thanks everyone.
Okay, thank you.
Thank you very much for coming.
Thanks.